Okay. Hi everyone. Thank you for joining ACROBiosystems webinar, Building the Perfect Antibody-based Therapeutic from Selection to Engineering and Manufacturing. We're really excited to dive into some of the most advanced techniques in antibody-based therapeutics. So today, we will discover how cutting-edge approaches, including the AI-driven candidate selection, optimization of antibody sequences, and also the conjugation method, and how they can help to address the key challenges in development of the therapeutic antibodies. So our speakers will guide you through the latest innovation in the antibody engineering. And let me introduce our speakers of today. So first, we will introduce Dr. Linglan Fu, the associate scientist at Zymeworks. And second, we will go with Dr. Ethan Lin, who is a senior scientist at Aureka Bio. And lastly, we will welcome Peter, our marketing product manager from ACROBiosystems. So we will go from this agenda, and we will have a Q&A section in the end. So let's welcome Dr. Linglan Fu to discuss how Zymeworks is optimizing antibody sequences to develop next generation therapeutics, including the boundaries of the therapeutic development. So Linglan, please share your screen so that we can see your slide.
Sounds good. Thanks, Brandy. Is it possible you can stop sharing?
Yes, I think I already stopped.
Hope everyone can see my screen now and
Yep, we can see
hear
it.
me okay? Okay, sounds good. Okay. Thanks, Brandy. And this is Linglan Fu from ADC Department of Zymeworks. I currently lead a production and biophysical characterization team supporting early stage discovery of antibody for our ADC team. And it's my pleasure to share our ADC development work here. And today I'm going to talk about a method development using capillary isoelectric focusing for high throughput dimidation quantification of antibodies. First, I will briefly talk about why dimidation of antibody could be a concern, how to monitor antibody dimidation, and current characterization methods and their limit. Then I will introduce a high-resolution CIF method that we developed for dimidation screening of antibodies. In the end, I will show a developability application of this method to monitor antibody chemical stability. Early screening of antibody molecules based on developability, biological functions, and efficacy and safety allows for a streamlined CMC phase. This involves careful assessment of developability parameters combined with binding affinity and biological evaluations. One of the major developability concerns is the chemical stability of antibodies, typically caused by post-translational modifications. And many chemical modification or degradation process could occur in antibody due to the nature of its sequence or storage conditions. This chemical modification may lead to product heterogeneity, change in biological functions or half-life, or immunogenicity of antibody products, and possibly impact on drug safety. So it's important to monitor this chemical modification in early stage of antibody development. One of the unfavorable post-translational modification is dimidation. Dimidation is when asparagine dimides into aspartic or isoaspartic acid, resulting in change in charge. Asparagine that close to glycine, serine, and histidine usually tend to have dimidation. In addition, storage conditions such as pH or temperature could lead to dimidation as well. So due to this change in charge, if the dimidation site on, for example, antibody CDR regions or the FC receptor binding regions, it may affect antibody binding to antigen or FC effector activity. All of these could potentially impact on biological function or efficacy of therapeutic molecules. So our question now is, how do we monitor or quantify demidation in antibody? Here are some listed methods that have been used for demidation detection or quantification. First, in silico prediction of amino acid sequence, which helps locate asparagine that have higher tendency of demidation. Experimentally, due to the changing charge and hydrophobicity, IEC, CIF, and HIC can be used for such characterization. However, when you have a stress sample with many post-translational modification, it could be challenging to identify the target demidation species. So currently, mass spec-based peptide mapping is the most reliable method for demidation detection or quantification, but data analysis can be time-consuming. So based on the charge shift caused by demidation, so here we develop a high throughput and high resolution CIF method to identify and quantify the demidation level of candidate antibodies. So what exactly is CIF, capillary isoelectrofocusing? CIF separate protein based on their isoelectric points or PIs. So here is the graph of the principle. So in the CIF cartridge, anode and cathode create a linear gradient of pH. Under an electric field, the protein migrate along the electric field until they reach the pH corresponding to the PI, where the positive and negative charge of the molecule are balanced. After separation, protein can be quantified by their native fluorescence or UV absorbance. So demidation of antibody result in an acidic shift due to the formation of aspartic acid or isoaspartic acid. In principle, we should be able to identify a demidation peak when compared to a negative control map with no demidation. And CIF is a plate-based assay with easy data analysis steps. Based on this, we intended to develop a high throughput method for demidation quantification via CIF. So first, how do we identify demidation species on CIF? For method development purpose, we used an antibody that previously confirmed with a demidation site in a CDR region on light chain by mass spec peptide mapping. So we can see this is a mass spec result. After 14-day human plasma or buffer incubation, the demidation level or demidation percentage at this location could go above 30 to 40%. Which significantly affect our on-cell binding and some efficacy results for this antibody. So we want to see, for example, during our early stage discovery, we want to see if any quicker or faster screening method to monitor this demidation in different candidates. So first, we just ran native full map on CIF by comparing the full map in human plasma at day 0 and day 14. We hope to see an acidic PI isoform of the antibody from day 14. However, we saw this cluster of PI peaks. The separation resolution is too poor to confidently identify a demidation peak of a full map due to the presence of many possible post-translational modifications. So how to increase the CIF resolution and isolate the demidation species of interest? So first, we know the demidation of this antibody occur on the light chain, and we hope to exclude post-translational modification of heavy chain by digesting the antibody into smaller fragments. To do this, we use IDS for digestion, TTT for reduction, and urea to create a denaturing condition for better dissociation of the chain fragments. In addition, running digested and undigested sample allow us to determine which peaks come from the heavy chain and which come from the light chain. On the top is the antibody after digestion and reduction. where it gets broken into FC, light chain, and FD. On the bottom is just a reduction only, where it gets broken into heavy chain and light chain. So by comparing digested and undigested condition, the CIF peak remain unchanged should come from heavy chain. This will help us identify peak species. In addition, we cloned adimidation positive control and negative control maps. First is N2D mutation, a asparagine to aspartic acid single-site mutant to mimic full dimidation as a positive control, and also N2S, which is asparagine to serine, was used as zero dimidation control. So we expect that first, the dimidation light chain peak will have a acidic shift from the white type light chain. And also we hope that we're expecting that the dimidation light chain peak of the white type will overlap with light chain peak of the positive control, which is N2D mutant. Here is our experimental setting. Fresh MAP1 sample were used, including its white type, N2D positive control, and N2S negative control. Samples were run in both digested and undigested conditions. So first, we use CESDS under reducing condition to confirm the digestion, IDS digestion went to completion. So after digestion and reduction, we expect to see 3 bands around 25 kilodalton because IDS will cleave below the hinge region and DTT will break the disulfide bond, leaving us the FD, FC, and light chain. Looking at the result on the left, there are 3 bands corresponding to the 3 antibody fragments. And there is a faint band around 60 kilodalton. You probably might not be able to see, but it was confirmed to be IDS itself by running IDS blank. So therefore, our digestion was successful with no remaining intact heavy chain. So using this fragmented sample, we got better PI separation resolution. And now we can identify heavy chain and light chain peaks on CIF. And here is the CIF graphs for the white type, undigested, digested, as well as positive control, N2D mutation, undigested, and digested. I will start off by describing how we identify heavy chain peaks. So any peak which change after digestion should come from heavy chain. So as you can see, the area below PI 8.4, all these peaks between digested and undigested, all these peaks differ, meaning they possibly come from heavy chain. And note that there's a peak at 8.5, which is present both before and after digestion. To identify this peak position, we can compare it between white type as well as N2D mutant. And the peak is nearly identical between N2D and white type. And we know that the only difference between white type and N2D is the single site mutation on the light chain. So if they are identical, meaning they come from the heavy chain. So we know that everything in this black box coming from heavy chain. And then we want to, we move to the right side. So just look at white type undigested and undigested. Those peaks are different, so possibly coming from heavy chain. And we do see a peak on the right side, around 8.88, that remain identical before and after digestion, which corresponding to our theoretical expectation of the light chain. And in the N2D mutation graphs, we do see a peak profile look similar, but with an acidic shift to 8.6. So we further confirm that this area should be our dimidated light chain peak, and this 8.8 should be our wild-type light chain peak. After identifying the heavy chain and light chain, as well as the demidated light chain, we want to further quantify demidation. So a calibration curve was established for this purpose. N2D and Y-type were mixed to mimic gradient of Y-type demidation level. For example, 100% N2D can mimic 100% demidation level, and 50% of N2D with 50% Y-type should mimic 50% demidation. So the mixture will run in both digested and undigested condition. The fluorescence, the native fluorescence of the light chain peak was detected, and we found that the peak is much lower. The peak intensity is much lower after digestion. Possibly the peak, even at 8.7, contains some both heavy chain and light chain. So for clean calculation, we only used the digestive version for the calibration curve. So we plotted a curve based on the fluorescence intensity of the demidation light chain peak against the N2D percentage. The sample mixture were run by both peptide mapping and CIF, and we found that the peptide mapping result and CIF results are highly compatible. And here are some analytical parameters of this method with LQ of 8.3% and LOD around 2%, with accuracy more than 90% compared to both theoretical mixture percentage and peptide mapping results. In the end, we want to try if this method can help monitor actual stressed map samples. So to force demidate the Y-type, samples were incubated in a high pH matrix at 37 degrees for 3, 5, and 7 days. And then samples were tested on both CIF and peptide mapping. The samples were then digested and denatured and reduced for the actual run. Here is the CIF sensorgram of the stressed sample, and the light chain peak position here from day 3 to day 7 of incubation, we can see an increase of peak intensity, indicating the increase of demidation level. And the CIF method, the result was compared with peptide mapping result of the forced demidation sample, and we were happy to see that the CIF demidation value were very comparable to the peptide mapping result. And in summary, here we developed a CIF method that helped us to quantify the demidation level of MAP1 using this reduced and digested condition. And this CIF method was validated by peptide mapping with accuracy more than 90%. And also this is a play-based assay and will have the capability for rapid screening of demidation in antibodies. And we hope this method can be used to optimize demidation developability in the early stage of discovery and help monitor CMC process. However, there's still some challenge for this method. First, we have to individually develop such method, for example, peak identification for different demidation site or different antibodies. And due to we do need those positive and negative control to mimic different demidation level. And also we have used this method for plasma incubated sample, but it showed very strong background signal. So for this, to test plasma samples, this method development is still ongoing. In the end, I would like to thank 3 key contributors in my team. And also I would like to thank everyone from ADC for their support. And thanks everyone. That's my what I want to share.
Thank you, Linglan, for such a insightful presentation on the antibody engineering and how Zymeworks is doing to optimize the antibody sequences and pushing the boundaries of the therapeutics development. So next, we will like to introduce Dr. Ethan Ling, who is the senior scientist at Aureka Bio. So he will share how AI-driven techniques are accelerating the selection, synthesis, and characterizations of the antibody candidates at scale, significantly speeding up the therapeutic discovery. So let's welcome Ethan Ling.
Hello, everyone. First of all, I would like to thank ACROBiosystems for the chances for me to share some of the work that we have been developing in Aureka Bio. So today I'm going to talk about like not just the concept, but how do we pushing the conceptual like idea into the actual application, like and some of the case studies that we have achieved based on this very exciting pipeline that could potentially facilitating the drug development for antibody like drugs or like potentially other specific targets like small molecule as well. So today I'm going to focusing on how this autonomous evolution, high throughput functional screening, and AID novel design, how does this like integration of these three modality will be facilitating the antibody discovery. So, yep. So as all of us know, conventional antibody discovery is low throughput and unpredictable, and it's a high risk business because you're talking about like tens of thousands, or even higher scale of the candidates that you have to screen out. Like probably like 95% of them it will be failing, and then if you proceeding with additional testing, in vitro testing, in vivo testing, then you're still going to be like eradicating lots of candidates because they are not fitting your PK profiles. It's having off-target effect, or its mechanism of action is not fitting your needs. And for example, like even though it's a high like yielding like an investment for time for lots of like therapeutics or like pharmaceutical company, but overall speaking, it's a slow process and it's also a high risk process. So I think the bottleneck here is that the screening is not really efficient and it takes a lot of time. So the really focus on this field is that how can we facilitate this and how can we de-risk like this all the candidates during this process so that we wouldn't spend a lot of time and efforts and resources on it and end up with something that does not like passing through like the whole clinical trial pipelines. So as we know that there's lots of like first generation biologics that's already in the commercial product market, such as like, the anti-HER2 antibody and some of the PD-1 blockade like biologics and some other like soluble protein that's targeting like liquid cancer that's also available in trial and also in commercial product as well. But nowadays, people are looking at the next generation biotherapeutics that is a little bit more like targeting, like driven. And for example, they are looking at multi-specific antibody and antibody drug conjugates or TCR engager, and or they're like looking at more of the hard to target like modalities such as like GPCR, ion channel, promiscuous like receptor or ligands, and or they want this potential antibody conjugated drugs to be activated at certain conditions, such as like pH sensitive, or like it's like triggered by tumor microenvironment, or like they're looking at some drug delivery that's a little bit complicated, such like it has to be passing through blood-brain barrier, so on and so forth. So there's more of the mechanistic part and also more of the criteria that has to be fit during this the initial drug screening so that it would not fail because now you see the threshold is like getting higher and higher. So in order to facilitate that and also overcome some of the burden, Aureka Bio is like trying to focusing on some of the modality that is going to be really facilitate this process. So for us, we have three different main modality that's being introduced here, and all start with something it's called let me pull out the pointer. It's called Aura Picasso, which is our generative AI model, like for designing the ligands. And it's not just stopping at AI because I know AI is a catchphrase these days, but not only we start with AI, then we go with this high throughput like synthetic biology based antibody discovery screening. like basically you can regard this as a very, very fast affinity maturation, like in the test tube. And then after like further screening out like some of the candidates, then it further goes into a cell-based like assay. So we have this like proprietary high throughput, high content functional droplet-based single cell screening that's not only being able to like testing the cell, like testing the functionality of your candidates on a cellular level, but it's also being able to like isolate and enriching those good candidates so that it can be either like progressing into additional testing for your candidates, or it can be further screening out or feeding back to the AI model for more refined studies. So let me get into some details of what is the modality that we're talking about here. So for the first modality, Aureka is the intelligent immunotherapeutic design system. So it's coming with three modality here. And this three modality is not like just like individual modules. It actually talks to each other because it's data rich like world these days. So all this like different piece of data, as long as we can intercollect them together, is going to be forming like a very, very useful model to further de-risking the design process. So the first module that we're looking at is structural modeling. So we are looking at the macromolecule like interaction. And you can getting affinity score. You can getting like how what is the conformation changing? What is the energy level? Like in terms of all this like molecule like protein interactions modeling. A second of it is that our de novo design focus system that is based on whatever functionality you are looking at. We are potentially this model can be giving you a set of like a whole big set of library sequences that can be catered to that potential binding affinity that you're looking for, or like you're looking at like some specific like pH modality. You can also potentially do that. And finally, the last part is the fitness prediction here. So fitness prediction is because even if you have an antibody that's all like seems to be binding well, that's just the beginning of the problem, right? Because you should be able to not only manufacturing it, putting conjugates on it, and then you must have like good stability and good solubility. And what about the cross reactivity? You don't want to like cross react to like something that you don't want. So this fitness prediction model is also like potentially serving as well another layer of de-risking. So it's considering like what like all this affinity or immunogenicity, solubility, cross-reactivity, so on and so forth. So at the end, the de novo model that the de novo sequence like for the screening library that's being designed is incorporating all these three modules, and then this was creating a foundation model that is using a large language model to generate all this library sequence of interest. So that's basically the whole idea of this AI centric like library generation process. But what's the differentiator here? Because there's also lots of like AI based like structure like models that's out there. I think our capacity here is that all this the module that I'm talking about is not just like a stiff model, like just like you just let stay as is. It's adaptive over time, especially as you can see, there's a three module. We are not only just using an AI model to generate the de novo library, we are also testing and fine tuning that by all the experimental data that is feeding back into the AI model. So at the end, the model that we have is adapted, is progressing over time so that your performance, your accuracy of prediction is getting better. So I think that's really going to be differentiated, especially if you're looking at the particular type of the drug development. You can think about this as like a long journey, like you are having a Gen 1 product, Gen 2 product, so on and so forth. And with this AI is also getting like evolve with the progression of your development, so you can get a better and better library in the future. For our quest, it's basically trained as an immunologist. We know that naturally in the human body, the affinity maturation of B cell, sometimes it takes weeks, if not months, like in your spleen, just be able to really like screen out the high affinity antibody over time. And of course, like that is definitely a bottleneck if you are trying to do that the traditional way in the drug screening. So this oral quest is that we are using the yeast expression system to expressing the ligand of interest, and also like potentially using that expressing the antibody of interest or ligand of interest, and detecting and the binding affinity through flow cytometry and sorting out the good binder, and then using this like good binder and passing the cell to diversify. And during the time, we are introducing some of the error-prone DNA polymerase so that we are basically facilitate the mutation inside of these E cells. And then we are basically facilitating the evolution of the sequences. And then after that, we express it, and then we do over this like a screening over and over again. So this basically facilitate the phenotype maturation process of that natural selection. And at the end is that we are being able to like achieve a way faster turnover rate. Like, so you're looking at 3 weeks of work instead of like 3 months or even more. So this really like facilitate a lot the process like in a short amount of time, and you also generate a very vast amount of data to train AI. Something that we found pretty useful is that because, as you can see, like in this kind of screening process, it's really important to have a consistency. So the consistency of this is that you must have a good recombinant protein or good recombinant antibody so that you can detect, and having the consistency over this rounds and rounds of screening. And particularly for our process, we use a lot of ACROBiosystems product on it, and it's like top-notch performance and having the consistency. Highly recommend that. So just in case if you haven't like tried their recombinant protein or antibody, give it a try. It may be worth it. And then the third like platform that we're looking at is Aureka. So think about like traditional, if you're doing a cell-based assay for screening, you're looking at lots of 96 flow plate, even you have a robot. But the problem is that that's a lot of plates, and there's also a lot of time or a lot of investment into like your robotic system and automation system. So what about it may sound like a dream, but what about I'm telling you that if you can potentially like combining all this well into a single droplet, like every single droplet is basically a well, then every single droplet that you're having here, you have your therapeutic library cells. You can see subtreating library, you can see like some small molecule or like some ligands of interest. And then you have your reporter cells that is like potentially having the reporter like different surface antigen or like different, you have a reporter like a construct like being built in. And then you can potentially using this to string out like whichever library that's really responsive and triggering the mechanism of action that you want, like in this like reporter cells of interest. So at the end, you're looking at a cell-based functional analysis that will save time, saving cost of goods, and most important is that it's also like provide a very big scalability in data generation that's capable of, of course, feeding back into any of the AI-based analysis. So I know that everything sounds like a dream, like right here. It sounds like very complicated, but you're saying that, oh, like it's maybe just a concept. But in Aureka, we actually make it a reality already. So we already have been using this like different modality on this platform, like in very integrated manners for different projects of interest. That, for example, we have a project that has been done on undruggable GPCR or discovery of pH-sensitive antibody, and looking at polymystical like receptor like that is what's the drug screening and bispecific ADC screenings. So I'm going to go in through some of the case studies that we have achieved with our partner. So let's begin with Aureka. Like let's take a closer look at the system. So like I say, you have your functional reporter cells of interest that can tell us like whether your mechanism of action is being achieved, like what based on your target, based on your drugs, like mechanism or your potential interest. Then you also have your library cells that's expressing your biologics. Then we are basically encapsulating both of them in so that like every single there's only one library cells and also like one or multiple like reporter cell depends on your assay into the single droplet by our microfluidic system. And then after that, like we of course like if there's additional reagents or like some other like additive that is totally depends on the assay design. And once we generate this like a lot of the droplets, then based on your assay window, we can incubate this droplet or we can just like run it right away. And then we will be running on our Aureka system, which is a microfluidics like based high content imaging system that you can potentially looking at for example, what is your like a reported cells like activity, and it is semi-quantitative, so that you are able to see, like, first, it's not no longer a yes or no question. It's going to be see, hey, I want a high responder, like or like do I want a fast binder? Do I want a slow binder? Do I want a higher responder, or do I want a delayed responder? So that's a lot of like opportunity here. You can screen out based on your assay design, and also based on your interest. So at the end, we are talking about this kind of screening method that's can screen through millions of droplets like within hours. So for the first case studies that, of course, this is very popular topic that is discovering CD19, CD3 bispecific T cell engager. So instead of going through the traditional method, we're trying to like benchmarking how fast can we do it. And at the end, we found that we can screen up like up to 1.5 million variants of library cells within hours. And then basically from start of this like library and the constructing of library to finish all the screening, and it only takes like 8 weeks. As you can see that as the candidate we screen out, like we can pick up for like potential functional assay, that we can see that is actually having pretty good like consistency, like and a very good performance compared to the wild type control here. And additionally, not just like the T cell engager is something that we can already do with the Aureka system, that we are also enable additional modality based on our high throughput screening, then can be used for screening for biparatopic antibody. For example, for this biparatopic antibody, we can be of course, it can be just like conjugated ADC, and you can also, by using that same method to screen for bispecific lysosomal degrader, or like I say, the bispecific ADC is also a possibility as well. So for the case study two, that what we do is that we are basically demonstrating that we are able to see like this anti-HER2 bispecific antibody, zanidatamab, that is already in the clinic, that we are able to see that having very strong binding affinity and also internalization of this particular antibody inside of our Aureka system. As you can see here, so these are the quantitative like data based on intensity of this internalization. And then this is the actual droplet. So we are able to see that the antibody is not only being able to be internalized, and we also see like very strong response as well. So just think about that. If this is going to be your drug product, your candidate based on the droplet, every single droplet that you are seeing here, you can sort it out and you can analyze the sequences. And these sequences can be feeding back into your data model of interest. So I think that's the power that's in present, and also like very creating lots of like efficient, like in terms of your data, like mining for the future. The third case study is more focusing on this like antibody design and also the screening that's facilitating based on our Quest and our AI model or a Picasso. So our objective, the challenge that we face from this particular project is that we're trying to improve the KD of this antibody to the picomolar level. And most importantly is that we want to screen out for the antibody candidate that can not only bind to target A, but also B. And both of them must have like relatively high affinities. And our solution is that we're incorporating our AI model and the structure-aware modeling, try to generate a very good already being de-risked like a set of libraries, and we can using our autonomous like affinity maturation models to really push it forward and verifying like what is the good binder, a bad binder, and the further testing it. So the further design looks like this. So we start with the AI design combinatorial library that is based on the antigen antibody structure. So you already have because we have all this like the protein binding models out there, so we can generating the library that is simply based on this prediction here, and then out of this, then we can then progressing into our Aureka system, so that they will be stringing out all this the binders that's having low binding affinity all the way to high binding affinity. And out of this, like because now we can sort it out, we can analyze the sequence, then this can be further feeding back into the AI like models so that you can see that, hey, so what is the sequence similarity? Is there any niche? Is there any similarity for the high binder and low binder? And after this being feeding back, it goes back to the AI and then generate a new set of libraries. So we keep on refining the library in a very, very short period of time. So that is really like reducing the bottleneck of the initial screening and de-risking as well. So at the end, we're able to screen out lots of the candidates that is that by based on the model that looks that's very promising, but when we are actually testing it, we see that, hey, some of them we see like some like goodbye some binding affinity, some of them don't. The most important is that we screen out like certain candidate that having very strong binding, not only to target A but also to target B. And with that, we can proceeding with this target. And by this time, the candidate pool has been like going from like tens of thousands or if not million scale all the way down to like whatever you can actually it's more manageable on the further validation assay. And so as you can see, like for our candidate, it's already showing that that's at the end it's being achieving that with the target AKD is definitely in the picomolar range. And then compared to wild type, we can see that the candidate we picked is actually having the performance that's tenfold or to 30-fold improved affinity compared to the wild type controls. The most important is that these are not done like in years. These are done within 3 months. So isn't that impressive? So at the end, I think I also want to give a step back and looking at the broader like overview and how this system can be incorporated together. So to recap, you think about like this over Picasso models. It's AI based structure aware de novo design of your library cells that is basically providing the maximum amount of the de-risking for you, based on this not only on structure wise, so it's also looking at the how like solubility and all this potential like potential hurdle you may face, like downstream of your like a drug development phase. So these are like the AI model is doing the maximize de-risking for you. And then once we have the initial library design, we quickly refine them by the Aureka system. And then by that, we have like some understanding of what is the good binder, what is the bad binder, and what really tells the differences in sequence there. Feed it back to AI model so that it goes on in a very fast pace, very short turnover time. And at the end, we can refine certain candidate we are confident to moving onward to the more time consuming and more resource consuming, like a screening part, which is always the mammalian cells part. So with that, we can use our Aureka system, like with the very dedicated, like a very fine-tuned like assay. So we can really screening out this candidate that is having the right mechanism of action for your antibody drugs, so that you can not only doing the screening, you are also de-risking because typically you have the target and you do a de-risking after it. So these are screening and de-risking all at the same time, high throughput and saving a lot of like cost of goods as well. At the end, you're going to get your candidates and you can further feeding it back to AI-based model for structured probability filtering. You can further be saying, hey, I know that this like several candidate now is fitting our mechanism of action requirements. So I just want to have an additional sanity check before I move on to on the CRO, like for some additional, in vitro and in vivo testing, which is very expensive. So we can really like feeding back to the AI model and it can tell so, hey, this is sanity check. This is a scoring for your actual feasibility or developability portfolio here. Is it good? Like if that looks good, then let's move on. So that really is like doing the de-risking part, like doing the screening process in a very efficient manners. So to recap, Aureka Bio is providing all this like different platform that we have developed here. Aureka system is high throughput, high content functional droplet-based single cell screening. While Quest is an affinity maturation in a tube and can generate like very rich data. Or Picasso is the AI model that have like lots of de-risking factor built in, and also it's based on the generative AI models. And at the end, we have to demonstrate that all these three different platforms is being able to having a success in improving and accelerating discovery of this antibody drugs. And that with different modality, different requirement, different mechanism of action. And we have been established a partnership with large pharma, biotech, venture group, and CDMO already. So hopefully this information is useful to you. And if some of this may seems to be fitting your drug discovery pipeline or for collaboration, please feel free to reach out to Wei On or Jenny that's listed here. And that's all for my presentation. I'm welcoming any questions afterward. Thank you.
Thank you, Dr. Lin, for showcasing how AI is transforming the antibody selection process and helping our researchers to work more efficiently. And it's really incredible to see how the technology is advancing therapeutic discovery. So next, we will welcome our final speaker today, Dr. Peter Xue, who is the Marketing Product Manager at ACROBiosystems. So he will walk us through how ACROBiosystems is enabling precise antibody-drug conjugation using advanced tools to ensure the optimal therapeutic outcomes.
Thank you, Brainy. Can you hear me okay? Then I'll move on to my talk for sure. So again, my name is Peter Xue. I'm the Marketing Product Manager. So I plan to walk you through some ADC development from the supplier perspective. So on this topic, we'll go over a variety of different topics, including the landscape of an ADC and providing you with an overview, governing factors, and the main challenges from the ADC developments across the industry. Start with optimizing the antibody, improving the payload conjugations, evaluate the payload delivery, and end with some ADC solution that we can provide to the field. Let's start with the landscape of the ADC. So when we look at this entire field, speaking from the overview of the ADC industry, we're looking at $26 billion market cap. We are forecasting by the 2028 has a very great potential as we realize for precision medicine. It's really combining two key components, the maturity of the monoclonal antibody developments and also standard of care of chemotherapy, especially in oncology landscape. With design to deliver chemotherapy to the very specific tissue type. Now we also see in the literature is a continued growing field as well. And as a different type of cancer have been targeted by ADC from phase 1 to phase 3 and beyond. So ADC is really bridging the combined benefit of the monoclonal antibody with their specificity designed to stimulate or inhibit or block the cell signal pathways, with the benefit of potency chemotherapy disrupting the cell cycle and the proliferations. So in ideal ADC drug development, design should amplify the both benefit of the monoclonal antibody and the cytotoxicity payloads in a precision medicine manner by maximizing the on-target efficacy and reduce the limitation like off-target toxicity. So, but in reality, there are the entire field face a lot of different types of challenges in developing a very reliable ADC. Really reflecting on the graph you can see on the left, only 12 out of 267 ADC have been made to the regulatory approval. These challenges can include off-target toxicity, insufficient benefit to conventional chemotherapies, also DART. Heterogeneity can also cause inconsistent cytotoxicity effect. Rapid ADC clearance also can cause the poor pharmacokinetic, also mean the ADC is being metabolized too quickly by the body. On the right, you can see there are different types of governing factors for ADC activity from target antigen, like the abundant expression level in the tumor versus normal tissue, heterogeneity of the tumor, internalizations of the ADC and shedding. Next is the antibody itself can bind to the target antigen, like the affinity, specificity, serum half-life, and the effector functions. We also have factors like conjugation and linker methods. You can see it a little bit down there. Different choice of payload options and tumor indication like target expressions and the heterogeneity, of course, and the payload sensitivity and resistance. So a lot to consider. In this talk, I mainly want to focus on the antibody itself, the conjugation, how to optimize the antibody, and next, improving the payload conjugations, And third, how to evaluate the payload delivery. So we talk about payload and the linker. Right. So first challenge is optimizing the antibody and the target affinity. This is really coming down to understanding the antibody by screening and optimizing antibody selections, which you should consider a balance of the antibody affinity in order to select effective ADC candidate. Previous two speakers did a good job talk about that already. So in our perspective, this might be different than how to select antibody drug. Their study has shown lower affinity can result in insufficient binding and the internalization at low target antigen density, which means they can cause off-target toxicity. In contrast, if you have a high affinity antibody, it can cause reduced receptor occupancy, which lower your efficacy. Therefore, we always suggest methods to balance antibody affinity to maximize the on-target toxicity, of course, and to mitigate the off-target effect. This all means you probably need an extensive screening campaign or strategy to find the best candidate by performing the technique like SPR, BLI, and even mass spec and other techniques. So I'd like to introduce a first case study, which utilize the high throughput SPR methods for screening and improving the antibody screening. This is a collaboration between ACRO and Actera. By performing a high throughput screening with our consistent recombinant protein like HER2, mesothelin, CCR5, and other difficult transmembrane protein, we're able to screen antibody candidate of your choice. Below is just showing the data. You can see among the data can be generated by screening transmembrane protein CCR5 receptor against 92 different antibodies. On the other side, we also, the case study number 2 is looking at FCR assay panel. So FCR revaluations is another important part of the antibody screening, of course, and really specialized helping you understand the ADCC, ADCP, and other functions. It sheds some light in an insight on the effective co-mechanism of actions for upregulating immune cell activity, antibody half-life, prolonging the PK and the efficacy. This is on the collaboration that we have done with Actera and also Dragonfly Therapeutic. So next, we'd like to look at the payload conjugations. So there are many different methods in the market in conjugation payload with the antibody. So we see there are challenges in terms of conjugation inconsistency with some of the conventions methods that are available. In contrast, we see a site-specific conjugation using glycosylation or glycoconjugation have produced a more homogeneous ADC. You can see use the laser. Yeah, you can see here. This helped the predictability of the ADC in preclinical and clinical setting. Other challenge that we see is adopting new payload. Even MMAE has the largest percentage of a current ADC drug. But we see the array of different type of payload with different mechanisms of actions. So in the next generation of ADC, we're not just limited to 1 to 3 major payload, perhaps. So there's a challenge on how to account for those potential variabilities or conjugation different type of payload as well. So at ACRO, our solution is providing the field with a universal payload conjugation kit that has a capability to conjugate a wide variety of different type of payload. So you can see that the top, the 2A. So our kits is one-step methods, either directly conjugate payload like MMAE or it could be a other type of tag like biotin with your antibody. Another method is adding the tryptophan or DBCO linker. Subsequently, you can add the payload of your choice with either using AZI, the click chemistry, or TCO to conjugate the antibody. Overall, either method give you a site-specific methods to produce a homogeneous ADC with payload either with DAR2 or DAR4 manners. So next, I'd really like to show you some data. So we would like to evaluate the performance of the ADC as a proof of concept. You can see using our AG-Link conjugation kits. we can add, we use the trastuzumab as a testing antibody that we conjugate the MMAE DAR2 or DAR4. You can see we're able to produce very homogeneous species of the antibody with the MMAE payload with a very well-defined peak. You can see in the HIC, HPLC analysis and show our conjugation kits is for sufficient and usually around 90 to 95% conjugation rate. Another thing that we test is the stability of the ADC in plasma over 8 days and observe ADC were stable in the vitro. This means that if we end up putting the ADC into the patients, ADC would not degrade in the plasma, but once it will be digested by the specific cell of interest and actually degrade inside the cell. So as you can see in the second figures. The last experiment that we like to show is whether this antibody aggregate during the conjugation, which is one of the biggest concerns that we observe. So we're able to see in our hand, we observe the ADC peaks are very well defined and had no obvious aggregation appear on during the experiments. So usually less than 5% are being aggregated, and very able to see in this graph. So besides looking at the biophysical or the biochemical characteristic of the ADC itself, we also like to validate their biological activity and the cytotoxicity. We first compare the binding of the HER2 antigen with this trastuzumab ADC with an antibody itself. We saw no differences in terms of binding activity, which means the conjugation process doesn't affect the antibody itself, which is very good to see. Next, we're looking into the cytotoxicity effect. So we'll put these two ADC and the antibody in Excel without the payload into the HER2 positive and also the HER2 negative cell lines. We saw ADC reduce the cell ability in the HER2 positive cell, but not the negative cell. And also the antibody itself doesn't do anything either. So the third challenge I'd like to introduce is the payload delivery. So another biggest challenge in this field is the linker cleavage efficacy. So you would like this to make sure the linker could be released the payload once inside the target cell. That's the point. And the enzymatic cleavable linker are commonly employed in the dipeptidic linker targeting the capsins or upregulated in many cancer types. Common enzymes like capsin B, NMP, and PLAU are commonly seen in the field. So next case study we're looking at is addressing how to test the linker cleavage efficiency in vitro. So we use the trastuzumab with a CX linker and the DXd payload. Capsin B is commonly upregulated in cancer cell, like we mentioned. So we add the ADC to the capsin B under the right environment to evaluate the linker cleavage efficacy. ADC DART value assessment by the LCMS here before the capsin B digestions, and after we observe some loss of a payload after the capsin B digestion. So in this experiment, in conclusions, ADC have very low cleavage efficacy. This gives us an example of how to evaluate the linker efficacy in vitro and can possibly predict a similar result in vivo. Another major challenge is how to evaluate the ADC in vivo PK analysis. So ADC is very hard to evaluate its pharmacokinetics due to the different aspect of the ADC itself. So since you are looking at not just the total antibody, but also conjugated antibody, ADC, conjugated drug, and total drugs. To model PK or PD of the ADC require many different analytical methods like the ligand binding assay, LCMS, and more. Lastly, it's require very precise measurements of PK and PD data when dealing with diverse patient populations. So last week, we can see there are different types of analytical methods, especially evaluating a larger molecule like ADC. The ligand-based assay, or the LBA, what we call it, is still a main methodology in serum tissue homogenate when we're looking at payload delivery in vivo. The figure here shows LBA using the anti-DXd against the ADC, but we're only able to detect the payload by this method. However, more advanced methods like LCMS allow us to monitor the free payload, smaller ADC fragment, and also still on the small portion of this being detected. Ideally, our customer would like to see a method that have a more advanced LCMS to detect different type of peptide species as well as the intact ADC to have a full picture of a different component of the ADC. But you can see you can imagine this is very challenging in the method development and hard to replace the traditional LBA in front of the PK at the moment, which has come to ADC antibody. So to wrap up my talk, I would just like to introduce our audience to some of the solution from ACRO in terms of ADC developments. So there's a lot of different products that we are offering from ACRO, starting from recombinant protein for antibody developments, even the recombinant antibody and the enzyme I mentioned. But we also do a lot more providing different kind of kits. This could be multiplex or singleplex kits, ADC conjugation kits that I introduced you, and also for manufacture of any residual detection kits as well. We also have a product line focused on med beads, our analytical surface and cell line surface as well. And if you have more interest, I'm happy to discuss with you more. With that, I just want to quickly mention what ACROBiosystems, why you should a better quality, better support, better response, and better design in terms of our product. We are very flexible, have good quality product. We're very customer-centric with good communication, and we have ability to scale up in your needs. So even from discovery, when we move on to preclinical, we're able to scale up based on your needs. We have a GMP facility now that can be accommodate for a higher quality, lower toxicity or endotoxin product for you. So antibody sample request. If you are not sure what you're looking for, the second QR code lead you to our website for the ADC developments, and the third one can download some ADC brochure from ACRO. So thank you for your time. I will take any questions as well.
Thank you, Peter, for this excellent presentation. So next, we will open the floor for a Q&A section. So we actually did receive a few questions from attendees. I'll go through them by the order. So first, question from Lilo. He's asking for the recording. Yes, we have the recordings ready, so we will send to all the attendees later after the webinar. Next question from Kyle. He's asking for Dr. Ethan Lin. Could you share more about the wet lab work associated with the Aureka's platform and how you validate its performance? And also, what downstream studies do you perform to validate their function?
Sure. Thanks, Kyle, for the question. So for the Aura Picasso platform, I assume you're also talking about the Aura Quest as well, because these are like basically tying pretty nicely together. So for the downstream performance validation, because we are talking about like when we are running that by the flow cytometry analysis, we can see that how well is the binder, and of course like there are some experimental design or like assay design that part I think is still proprietary. our AI model, is it a good fit or not a good fit. Additionally, is that when this goes into the overall quest assay, which is a cell-based assay, then we have like such as like flow cytometry assay to really testing that, first of all, is it binding, and also how does it have the right mechanism of action. And then the next thing is that we are also having some microscopy assay in development as well. So can testing that if the antibody is really get into the cell, like by the method that you want them. Yeah. Hope that answered your question. Yeah. Yeah.
Yeah. We will move to the next question. So from Kenyo. So the question is, the development ability portfolio is fascinating. Is that method very specific to antibodies?
So far, I think as far as I know, it is more catered to antibody, but I do think that the model, especially like if you do have the data for small molecule or something like that, that's also something that we can potentially train the model on. So I think there's definitely a possibility that this developability like model or the module can be extended to other application as well.
Thank you for the answer. Yeah. So we do have two more questions are from Lee. So the question is, number one, could you explain a little bit more of the application of Aureka's platforms on the nano design? And number two, how's the experimental results or data backflow to your AI model? Any concerns of the data quality and correlations?
I mean, so far, I'll answer the second question. So first, so far I think we are pretty satisfied on our AI model's performance, but because we do see that, like based on the refined AI model and based on the refined candidate list, we're able to select an antibody candidate that is showing good binding affinity and also having the right mechanism of action that we want them we wanted to see. So I would say the performance so far is already pretty good. Of course, like I say, the AI model is really adaptive. So as we speak, we are still generating more data to feed back into the AI model. So I think the performance will only get better. So in terms of the first question, the application of Aureka's platform on the novel design. So I think for that particular part, I'm not quite sure, like because for the novel design, it's basically depends on your target interest. If you, for example, if you are looking at like such as ligands binding to specific receptor as an agonist and or antagonist, that's something that can potentially design differently, right? And also, if you're looking at like a bispecific binding, or you're looking at biparatopic antibody binding onto the same receptor, like or like you're looking at two different molecule, we can also design like based on that as well. So, or if you're looking at some even like more complicated, like binding scenarios, such as like you're looking at nanobody having certain like pH sensitive like capacity, and but you still have to retain the binding affinity at that pH range. Something like that. I think that's also the potential way that we can have this de novo design application to work. Yeah.
Thank you so much, Dr. Ling. Any other questions? Feel free to raise your hand and or type your question in the Q&A sessions. I don't see any more questions at this moment. So I'll just wrap up quickly. So thank you so much to all our speakers, Dr. Linglan Fu, Ethan Ling, and also Peter Xue, for their informative presentations. And thank you to everyone for attending today's webinar. We hope you found the content very valuable and insightful. So if you have any further questions or you would like to follow up with our scientists, feel free to reach out to us directly, and we look forward to connecting with you at future events. So have a good week, Ken.
Thank you.
Thank you.
Thank you.
Thank you.